I'm Daniel Almonacid. I'm the head of product development and data science at a small startup here in Sacramento, California called Biomakers. And hopefully by the end of these 10 minutes, you will know more of what we're doing related to soil health. So the title of my presentation today is how we can assess soil health using soil microbiome analysis. And good news is that in the previous presentation, you already saw a map of France measuring the microbiota that was present in the soil. So that was a very helpful introduction to what I'm about to present here. I wanted to remark the following before I show you how we analyze soils. The USDA defines soil health as the capacity of the soil to function as a vital living ecosystem that sustains life. Soil is a symbiotic ecosystem that can be managed to provide nutrients for plant growth. So based on that definition of soil health, we would like to argue that measuring soil health is directly related to measuring the microorganisms that live in that soil. And that's exactly what our startup here in Sacramento is set up to do. So I'm going to show you how we do that. Basically, to summarize, I would like to mention we're a scientific company, data-driven. What we are poised to do here is to understand what soils really need in terms of which practice or ag input you need to use in that particular soil. So then the fertility is increased and the production, but in a sustainable way. You also reduce uncertainty with diseases and so on. So the way in which we assess soil health, it's through a product that we call B-Crop. And basically, B-Crop is just, it starts with a soil sample. So basically, we recommend normally 3 up to 10 soil cores per hectare or the area that you want to plot. Then we recommend it being around 2 to 4 inches deep. So basically, you don't have the soil at the top, which is devoid of some of the microorganisms that don't tolerate oxygen. So we recommend going a bit further down. And then you can ship that sample to our lab in Sacramento if you are here in the United States. And if you are in Europe, you can send it to Valladolid, Spain, where we have another processing lab. And the process looks a little bit like this. We extract the DNA of the living microorganisms in that soil. We use sequencing technology to read the DNA that is present from those microorganisms. And then we go through a number of bioinformatic and informatic processing so we can provide a report both in PDF format and also through a web portal that is interactive. And what you will see in those reports are basically 3 types of information. One is taxonomy level information. In other words, which microorganisms are living there in your soil. We have a list for bacteria and archaea on the one side, and then for fungi. So we present those 2 lists of microorganisms and their relative abundance in the soil. Then we also look at functionality of the soil. And for that, what we do is, given the presence of the microorganisms that we know are in the sample and their known genomic compositions from databases, we can therefore come up with a functional inference of which genes are present in that pool. So with that information, we're able to understand what's the potential of that soil for a different set of functions that I'm going to go in the next slide. And then last but not least, we also look at ecological biomarkers. So what we do is that we have a species list. So we look at all of the bacteria that are present and archaea on one table, and then in a different table, we have all of the fungi. And what we do is that we convert that list into an interaction map, a network. And that network basically summarizes the interaction between different microorganisms, which microorganisms like to co-occur and which like to co-exclude one another. And based on that, we can derive some knowledge of the soil. And I'm going to show you now clearly examples of each of them. So this is how the report looked like. It has 3 categories. The first one, it's called biodiversity. So there we assess what is the diversity in terms of taxonomy, how many microorganisms are there, what is their abundance. Then we look at the functional diversity, which means which functions is the soil providing. Is it cycling nitrogen? Is it cycling phosphorus? Is it providing hormones that are known to be produced by the bacteria? Are they providing salt tolerance molecules and so on? And finally, you get to the resistance, which is a metric based on the interaction plots that I showed before. And I'm gonna go, I have a specific slide for that because I think that's very interesting. So I'm gonna skip that to the very end. Then we talk about health, soil health. So in the soil health component, we talk about the pathogens that we detected and how abundant they are in comparison with their with the microorganisms that tend to produce molecules that can counteract their effect. So basically, it's a health risk score that we provide in our report. Then in the next section, we have the biocontrol species. The hormones that are being produced in the soil, as I mentioned, in functionality and the stress adaptation molecules. And finally, we also talk about nutrient cycling. As I mentioned earlier, we talk about what's happening with the nitrogen pathways in the soil. Is the nitrogen being made available in plant available forms, or is it consumed by bacteria? So that's exactly what we look at in our report. And as I said, the last message that I want to leave here is interactions between microorganisms. So basically, this is how one of those interaction maps look like. And we have an article in which we were investigating vineyard soils from the US and Spain. And the beauty of this type of representation is that it's agnostic of location. So basically you can have a list of microorganisms that is very different in one location versus the other. But then when you do an interaction map, the interactions are agnostic to the label of each microorganism. So each dot here is a microorganism, and each connection means that these microorganisms co-occur more than one would expect by random chance in our database. And what you are seeing here is the same network generated for a conventionally managed vineyard, for an organic vineyard here in the middle, and from a biodynamic vineyard in the right. And what you can see here is that the topology of the network is very different. And actually, you don't even have to look at the network. You can measure properties of this network and come up with that score. And that's exactly the score that comes up in the resistance component of our report. Basically, we have been able to correlate the topology of these networks with type of management in vineyards. We have also subsequently been able to demonstrate that these interactions are very relevant for other interesting features such as yield and nutritional composition of the crops and so on. So that's me. Before I say goodbye, I just wanted to mention that Biomakers has an ongoing global soil health initiative called Fields Forever. The goal is to open up our technology to researchers, to scientists, to people that is working in the soil and want to take action in making soils healthier. We have a €2 million commitment from the European Union through a grant, and we also have many, many partners across the globe. We already have 100 ongoing projects in 4 continents. One of them is from the Urban Soils Institute, where we are sampling 540 different we will have 540 different samples that we're going to analyze and see what's going on with the efforts that you guys are making there. So thank you so much for the invitation to have us, and hopefully we have some fruitful discussions at the end of the session.